Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. This week we're going to be talking about the flow of genetic information from DNA to RNA to eventually the protein. So without further ado, let's get into this. Before we really jump into the main material of today's videos, right now the terms and definitions slide should be popping up around me so that you can grab the vocabulary we will be using today in this video. However, if you would like the full PDF version of my slideshow, please feel free to head on over to my Patreon. The link is down in the description below. And from there, you can grab all of the study material that goes along with this video for absolutely free. However, patrons will get that study material and this video at least a day early. Before we jump into the new stuff, let's begin with a quick review. First of all, the phenotype describes an organism's physical appearance, so everything that is actually expressed. And the genotype describes an organism's genetic makeup, so what the DNA actually says on those chromosomes. And if we remember, DNA is the heritable genetic material responsible for all of the genotypes and phenotypes that we see in life today. So the two main questions that we're kind of going to be answering here are, what is the connection between the genotype and the phenotype, and how does information go from the DNA where it resides and get translated into synthesizing a protein, because proteins are essentially what we see in the phenotype. So this information has to go from the genotype described on the DNA all the way into being expressed as a protein in that phenotype. This whole process of taking information from the DNA and expressing it in proteins has two main steps that we're going to talk about today. The first step is transcription, where RNA is synthesized using DNA as a template, and this happens in the nucleus. And the second main step is called translation, where that information on the strand of RNA is then used to synthesize a polypeptide or protein. And this process happens in the cytoplasm. So before we really talk about these two processes, we need to think of this whole thing as kind of the chemical languages of the genetic information. And the first chemical language is the language of DNA and RNA. And this language can be described by 4 nitrogenous bases that we talked about in previous videos. Well, technically 5 nitrogenous bases, because if we remember, RNA switches that T for a U. And then the other chemical language is the language of polypeptides, which can be described with the 20 amino acids used to make up all the proteins that make the life that we can see today. So the big question here is, how do 4 nitrogenous bases code for those 20 amino acids? So how do we go from 4 things up to 20 things? Well, that answer is because there's a thing known as the Triplet Code. And the Triplet Code kind of describes these sets of 3 nucleotide long words that will code for a specific amino acid in the polypeptide. And this 3 nucleotide long word is the codon. So on their own, each nucleotide, so each base, is exactly that. It's just one of the 4 nitrogenous bases. But if you get it in a set of 3, it's coding for something specific. So 3 nucleotides make up a specific codon. And you can have many codons along a specific strand of RNA. And that particular sequence that they occur in will result in a particular sequence of amino acids in that polypeptide and code for that specific protein. So we know these 3 nucleotide long codons code for specific amino acids. And if we start actually looking at what these codons code for, we get the genetic code, which is basically the rule book that kind of dictates what a specific codon codes for. And if we see here, we see the genetic code kind of represented in a circle chart that you can follow to figure out what particular sequence of nucleic acids will result in what. Now, I don't really like the circle chart. I had this actually introduced to me as a chart that looks more like this, which I think is a little bit more clear. So we're going to work with this chart instead of the circle chart that the book uses. However, you're going to get to the same result either way. It's just a different way of depicting it. So first things first, how do we read this chart? Well, the left side of the chart is going to be the first position. So it's going to be the first base in the 3-base sequence that we're looking at. The top row of the chart represents the second base, and then the right side represents the third base. So for example, if we're going to be looking at a specific thing in this chart, say the PHE or the phenylalanine that I have circled with the purple box, we'll see that on the left side, which is going to be the first base in the sequence that codes for phenylalanine, is a U. And then up top, the second base is also U. So far we have U, U. And then if we follow that row all the way across to that third position on the right side, we come up with a third U. So phenylalanine is U, U, U. U, U, U codes for phenylalanine. Whereas if we go somewhere else in the chart, like the GLN or glutamine I have circled with the kind of orangey yellow box, the first position, so the first base on that left-hand side is C. The second position, so the base shown on the top is, the second base is A. And then on that right-hand side, the third position, so the third base, is also A. So glutamine is coded for as CAA. And that's fairly simple. It's kind of like a multiplication chart where you're just following based on the row and column that the thing you're looking at is in. And then at the bottom, we have the chart that tells you what each 3-letter abbreviation amino acid is. But in an entry-level bio class, the important thing is going to be able to read the chart that tells you the genetic code. So if your professor gives you a particular codon and asks you what does this code for, you can look at the chart and figure it out. A few quick notes though, there are multiple ways to get a particular amino acid. So some sequences of bases will actually end up coding for the same thing. So as we can see here, just at a quick glance, there are at least the 2 ways in that very top box on that left-hand side to get phenylalanine. And if we notice, there are multiple ways to get multiple bases in most cases. Some sequences are what we call stop codons. So UAG, UAA, and UGA are the 3 stop codons. And stop codons do not code for a particular amino acid. They actually are a signal for translation to stop. And we'll get more into that when we get into the translation slides in a couple minutes here. And then there is one sequence, AUG, that is the start codon. And this is the codon that marks the place where the initiator binds and translation begins. And it actually does code for a specific amino acid. It just happens to also be the amino acid that kicks everything off during translation. And again, we will get into that in a little more detail here in just a second. Now that we've talked about the genetic code and you kind of have an idea of how we go from these 4 bases to the 20 amino acids, because it's not the individual base that codes for an amino acid, it is a sequence of 3 bases known as a codon. We can talk about the transcription of a gene. So step 1 in this whole process of going from DNA to protein. Transcription happens in 3 basic steps. Step 1, initiation, where RNA polymerase will attach at the promoter. Step 2, elongation, where that RNA strand will then grow. And step 3, termination, where RNA polymerase reaches the terminator and detaches. And I know I just said a lot of words, but we're going to get into that right now. Step 1, initiation. So RNA polymerase is the thing that actually is going to make the RNA. And what it does is it's going to attach at what is known as the promoter. And the promoter is just kind of a part of the DNA that signals the start of the gene. So that's where RNA polymerase knows to attach, because that is the beginning of the gene. And it's just a particular sequence that's going to be recognized. And so when RNA polymerase attaches to that strand of DNA, it's going to separate the double helix, and then one strand is going to be used as the template to make the RNA. And then the other strand, since DNA is a double helix, is going to go unused, and it'll kind of be held up out of the way in the other side of RNA polymerase. So step 2, elongation. Elongation is basically just where that RNA strand gets longer. It kind of names itself. Scientists aren't always that clever. But to get into a little more detail here, that new strand of RNA is getting synthesized. So the RNA nucleotides are going to briefly form little hydrogen bonds with the template DNA strand. And then the RNA nucleotides are going to be added to that RNA strand, and new bonds between them are going to form. And then as RNA polymerase moves along that strand of DNA, the DNA strands that have been separated will reunite and form that double helix right back up as if nothing ever happened. And then step 3, termination. RNA polymerase reaches the terminator DNA, which is just a sequence along the DNA that marks the end of the gene and where RNA polymerase can stop making RNA and can then detach from that strand of DNA. So then once that happens, RNA polymerase just kind of says, I'm done. And then the DNA strand can completely attach back to itself. And we end up with that completed RNA strand being released from RNA polymerase. So if you remember, this whole first process of making the RNA happens in the nucleus, and then the protein synthesis happens out in the cytoplasm. So how exactly do we get that new RNA strand from the nucleus to the cytoplasm? Well, first of all, that RNA strand has to be processed a little bit because it's not quite ready to be packaged out for translation to happen. The first thing that needs to happen is that RNA strand needs to get a cap and a tail. And the cap and the tail basically just kind of protect it during everything. And another thing that needs to happen, because this RNA strand contains both coding and non-coding portions, that stuff kind of has to be removed and spliced together so that we don't have a lot of junk in that strand. So these non-coding segments are called introns, and the introns are going to be cut out. And the coding segments are called exons, and those exons are going to get spliced together. And this whole process of the cutting and splicing is called RNA splicing, again, because scientists are super clever with their naming and super cryptic. So Once that RNA splicing happens, once this RNA strand is processed so that it doesn't contain a bunch of junk and it has that cap and tail on it to protect it, the end result is something we call messenger RNA or mRNA. In this video, we're going to be talking about a lot of different types of RNA, so just kind of keep that in mind, what each particular kind of RNA does. So mRNA is the messenger RNA that is actually going to be used to make that new polypeptide. And once that mRNA is finished being processed, it can then exit the nucleus and head out into the cytoplasm for the next step. Before we get into the nitty-gritty of the next step, let's talk about another type of RNA that's a big player in translation. And this RNA is known as transfer RNA or tRNA. And the easy way to remember what tRNA does is it kind of acts as the translator to go from that DNA RNA language into the polypeptide language. So the two functions that tRNA kind of carries out during the process of translation is that it, one, needs to recognize the specific codons on that strand of mRNA, which is the message from DNA to the protein building things. And it also needs to pick up the appropriate amino acid that goes along with the codon on the mRNA so that protein can be built in the correct sequence. So at one end of the tRNA, because during the process of translation, we will use many different tRNAs. Just keep that in mind. Each tRNA has at one end a specific anticodon. And basically an anticodon is just the complement to a specific codon. So an anticodon will recognize a specific codon. And then at the other end is the spot that will attach to a specific amino acid. So each single tRNA recognizes one codon and attaches to one amino acid. And so this slide here just shows a much simpler version than the computer model we saw on the other slide of kind of what a tRNA does. It has a flattened 2D kind of version, a little bit of a 3D version that shows where different hydrogen bonds are and things like that. Or on the very right, the version that you're going to see in most textbooks that most clearly represents what the tRNA is doing. So at the bottom there, we see the anticodon that's going to recognize the codons on that strand of mRNA, and the little purple thing at the other end that attaches to the specific amino acid. So now let's talk a little bit about ribosomes, because ribosomes are the things that actually build the polypeptides. And a particular ribosome is going to be composed of two subunits, the large subunit and the small subunit. And both subunits are made up of proteins and another kind of RNA, ribosomal RNA, or rRNA. And basically, the thing that you need to remember about rRNA is just that it's part of the stuff that makes up ribosomes. So what happens with the ribosomes during translation is that that mRNA strand is going to bind to the small subunit, and then the tRNA is going to bind to the large subunit throughout this whole process. So the small subunit kind of keeps that mRNA moving along, and the large subunit keeps the particular units of tRNA moving along and actually builds the polypeptide out of that. And so because those subunits are kind of attached like that during the process, the mRNA and the tRNA are held close enough together for the codons to be recognized. And then the amino acids are also held close enough together so that they can then be transferred from the tRNA to that growing chain of the polypeptide. So let's talk about translation. And one thing you're going to notice is translation also happens in 3 steps. And they're the exact same 3 steps that we saw before. Slightly different things happen, but they're called the same things. So that kind of makes it easy. So translation step 1, initiation. So the first part of initiation, that mRNA strand is going to bind to the small subunit, and the initiator tRNA is going to bind to the start codon, which remember is AUG. And the specific amino acid here that's coded for at that start codon is methionine, or MET. And the anticodon for that that's going to be on the initiator tRNA is UAC. You may or may not need to know that depending on any exams your professor will give you. So that is a very helpful thing to know. However, if you remember that the start codon is AUG and you know the complements, you already know what the anticodon is because you can look at that start codon and figure out what the complements for all those bases are. Then you just have to remember that the amino acid is methionine, MET. So then the second part that happens here during initiation is that the large subunit then binds to the small subunit, and that initiator tRNA will bind to what is known as the P site. And we will talk more about the P site in the next slide, but the big deal here, the initiation, is that mRNA binds to the start codon, the mRNA binds to the small subunit, the initiator tRNA binds to the start codon, and then the large subunit binds to the small subunit. And so if we have this kind of diagram blown up a little more, you can kind of see that happening. So kind of everything that happens with the binding on the small subunit happens sort of at once, and then that large subunit comes in. Because when proteins are not being synthesized, the small and large subunits are not bound together. So translation step 2, elongation. So this step can be a little bit confusing, but it's basically just a cycle. So an incoming tRNA anticodon is going to pair with the next mRNA codon, which is in what is known as the A site. In that large subunit where the tRNAs are binding, there are 2 sites, the P site and the A site. The A site is where the next tRNA comes into, and the P site is where the tRNA previously is at. And that will make a little bit more sense when we kind of blow up this diagram in a second. So once that incoming tRNA binds to the codon in the A site, the polypeptide that was held on to the tRNA currently in the P site is going to form a peptide bond with the amino acid on that new tRNA that's in the A site. And then once that happens, the tRNA in the P site is going to leave the ribosome altogether. Its job is now done. And then the kind of third step of this is that the ribosome is then going to move that tRNA in the A site to the P site. So if we look at this diagram a little bit bigger, on the top, that next tRNA with the next amino acid comes in and binds at that A site where the next codon is. So that's the codon recognition. It will then move over into step 2, where we can see this polypeptide chain is going to begin to detach from the tRNA that it's currently attached to and attached to the amino acid that came in on the new tRNA. And then once that happens, that original tRNA that was already in the subunit gets removed, and it goes on its way And everything after that must then shift down so that there is a new codon in that A site, and the polypeptide chain is then moved back to the P site. So kind of an easy way to remember what happens at each site is that the A site is the attachment site. So the A site is kind of where everything happens. The tRNA attaches in the A site, and then that polypeptide chain attaches to the new amino acid in the A site. And then the P site is kind of the polypeptide site. That's where the tRNA that gets the polypeptide chain attached to it gets moved to, and when everything is said and done, that's the site that the polypeptide gets sent out of, because that's where the exit site for that polypeptide chain is. So A site, attachment site, P site is just kind of the polypeptide site. Once that polypeptide is attached to the tRNA, everything has to move down to the P site. So then that brings us to step 3, which is termination. So the first part of termination is that the stop codon is reached. So either UAA, UAG, or UGA. And then since stop codons don't code for a particular amino acid, they only signal for translation to stop. The polypeptide is then freed from the tRNA that was holding it in that P site, and it is just removed from the ribosome entirely. The polypeptide is created at this point, and then the ribosomal subunits will separate, and the mRNA will eventually be broken down. And so that's pretty much how it happens. Just those 3 steps that kind of keep going until that stop codon is reached, and then everything just kind of releases. Everything is done, and the mRNA, again, will get broken down eventually. So what happens when something goes wrong? Well, you end up with a mutation. Now, we will get into mutations in a little more depth in a future video when we talk more about evolution and the forces that drive evolution. But today, we still need to talk about mutations because they do impact how that flow of information goes from DNA to RNA. So a mutation is any change to the genetic information of a cell, and they occur specifically in the DNA. And because they occur in the DNA, they alter what is coded for on a particular strand of RNA. Since, remember, the flow of information is DNA to RNA to protein. So we're going to be talking about it as one nucleotide, but remember, DNA is a double helix, so it has 2 strands. So we're really talking about a pair of nucleotides, but generally when you discuss mutations, you're looking at the resulting RNA, not the whole DNA strand. So that's why we're talking about it as one nucleotide. So a substitution replaces one nucleotide with another. It's a particular kind of mutation, and it just swaps a nucleotide. A deletion is a type of mutation that removes a nucleotide. So it'll take a nucleotide away, but it won't replace it with anything else. An insertion adds a nucleotide. So it doesn't take anything away, it just adds something extra. And then mutations, while they can just be like random whoopsies that happen because life is messy and things happen, they can also be caused by things known as mutagens. And mutagens are some sort of chemical or physical agent that has the ability to interact with DNA and change the genetic information there and cause a mutation. So before we wrap up here, let's take a brief look at these 4 main types of mutations that occur. So the first type is called a silent mutation, and a silent mutation always is the result of a substitution. So it's called a silent mutation because the resulting codon ends up coding for the same amino acid. Remember when we talked about the genetic code a few minutes ago, we mentioned that there are multiple ways to code for one amino acid. And because of this, if you swap a particular base and end up with a codon that ends up translating to the same amino acid, in the grand scheme of things, nothing has really changed. You still end up with the same polypeptide, but it is still a mutation because the genetic code has still been changed. There's still a different codon there. But the result is essentially silent. The next type of mutation is called a missense mutation. And this is where you get a substitution and the result is one amino acid codon is actually changed into a different amino acid codon. So instead of coding for the first amino acid, it gets changed into something entirely different, but it's still coding for an amino acid. It's just when the polypeptide is made, you're going to have the wrong sequence of amino acids, so it's going to be something completely different. The third type of mutation we're going to talk about briefly is called a nonsense mutation. So it's very similar to a missense mutation in that the amino acid codon is changed into something else. However, it's the amino acid codon changes into a stop codon. So instead of coding for an amino acid, you get a stop. So the polypeptide chain will be cut short prematurely. And then the last type of mutation is called a frameshift mutation. And this is caused by either an insertion or a deletion. And what happens when you get that insertion or deletion is that all of the nucleotides after that particular thing will then have to be regrouped because they're no longer in the same order. And this may result in many new codons. It may result in one new codon depending on where that insertion or deletion happens. So the frameshift mutation can cause a lot of trouble potentially. But that's all we're really going to say about mutations right now. Like I said, we will get into them more in future videos when we talk about evolution. So I know that this video has been quite a bit full of information today and it's been a bit of a longer one, but congratulations for getting this far because that's all I have for you today. And if everything went according to plan, there should also be a couple of like charts that kind of spell out what the different RNA do and what happens during translation and transcription for you. That will also be available with my PDF slideshow. So please take advantage of that if you need some more study material. And I hope you have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.